Hello everybody out there. Hello. Hello. Skeletal muscle fiber. Skeletal muscle fiber. Skeletal muscle fiber. Skeletal muscle fiber. Hello and welcome. Yes. Alhamdulillah. I got back into the settings. The video setting is proper. The audio setting is proper, I guess. If any problem with the video or audio setting, please let me know. I'll immediately correct it. All right. So let us begin our discussion. Today's class is basics of physiology. In this, we'll be discussing what is the structure and functional aspects of what are the structure and functional aspects of the skeletal muscle. The skeletal muscle, we'll try to first understand what is the ultrastructure. Then we'll try to understand what are the proteins present in the skeletal muscle. Then we try to understand what is the function of the skeletal muscle, how the contraction is occurring, relaxation is occurring. And we'll try to understand, apply the same principle on the smooth muscle and see whether it holds good or not. All right. So the picture in front of you, if it is clear, give me a second. I'll just change this. Yes. The picture in front of you is a skeletal muscle. It's a skeletal muscle fiber. A skeletal muscle fiber Hello. Yes. So the skeletal muscle fiber ultrastructure we'll try to understand. The picture in front of you is an excellent picture of a skeletal muscle fiber. This picture tells us that a muscle is made up of individual cells called as striated, the skeletal muscle fibers. So a muscle is a bunch of fibers. A muscle is a bunch of muscle fibers. These muscle fibers are longitudinally placed within the muscles, and each muscle fiber, they have got myofibril. So if you see this, or I'll try to highlight this, So myofibril, all right. The myofibril. So each fiber consists of multiple myofibrils. All right, what is the structure of myofibril? A myofibril can be subdivided into individual sarcomere. So each myofibril, you can divide it into individual sarcomere. Where is the sarcomere? I'll highlight it now. So this is the sarcomere. This is the sarcomere. All right. See, please follow this properly, because once you understand this, you'll understand the excitation contraction coupling. All right. So sarcomere is nothing but each of the myofibril is divided into sarcomeres. What is a sarcomere? It is a unit of excitation contraction coupling. A sarcomere is demarcated. A sarcomere begins with a Z line, ends with a Z line. So a muscle fiber, part of the myofibril, which is present in between two Z lines. Z line is a crisscross line. You are calling it a Z line because it's a crisscross line. So this line, if you see the Z line, if you see, it's a crisscross Z-like line. All right, like an alphabet. So this is the Z line. Are you following it? Yes. So each Z line demarcates beginning of sarcomere, and end of sarcomere is with a Z line. So from one Z line to another, the portion of myofibril, you are calling it as a sarcomere. Each sarcomere composed of filaments which create bands. These filaments are of two types: the thick filament and the thin filament. Thick filament, you are calling it as myosin, and thin filament, you are calling it as actin. Thick filament and thin filament. So thick filament and thin filament, myosin and actin. Each myosin and actin are arranged in such a manner that it creates certain zones and bands. It creates certain zones and bands. The contraction causes with the contraction of the muscle fiber. With the contraction of the muscle fiber, what happens is there is a H zone, there is a band, there is a I band in between the two Z lines, and centralmost portion is M line. I'll explain you what are these with multiple diagrams and photographs, pictures I have. I'll explain, but for now, please remember, I'm just introducing you a new term called as A band, I band, H zone. So they'll ask you, with contraction, will it change or not? The length of these muscle fibers, the length of these zones and bands that are present on your screen, they change or not? So H zone, the H zone and the I zone, the H zone and the I band, they actually change. They get shortened with muscle contraction. They get shortened with muscle contraction. But the surprising fact here is, the beauty of this excitation contraction coupling is that with contraction, there is no change in the length of A band. There is no change in the length of A band. A band does not change. The A band does not change. So we have got thick filaments and thin filaments. Thick filaments are myosin filaments, and the thin filaments are actin filaments. So in this next diagram, we'll try to understand what are the cross bridges, what are the parameters of a sarcomere. So if you can closely observe the central portion of the sarcomere, it is made up of the thick myosin filaments. The myosin filament has got short ridges or anchors. We call it titin. T-I-T-I-N. Titin. T-I-T-I-N. This titin anchors the myosin and is a very, very important component for muscles elasticity. It maintains the muscles elasticity of the muscle fiber is maintained by this trident. All right. So if you again observe this, we have got central, I'll highlight it. So this is the thick filament and this area is the thin filament. This is the thin filament. All right. So you can see the cross bridges or anchors. This is called as titin. All right. So again, a human sarcomere is nothing but multiple sarcomeres form myofibrils, multiple myofibrils form muscle fiber, multiple muscle fibers form muscle. I repeat, sarcomeres, then myofibrils, then muscle fibers, then muscle. So this is the chronology. You have to remember that the sarcomere is composed of thin filaments and thick filaments. The arrangement of this thin and thick filament makes the Z line, the I band, the A band, and the M line, and the H zone. All right. So let us try to understand what are these. The external and internal membrane system of the skeletal muscle is composed of the external and internal muscle. If you go from outside to inside, so say suppose these are the muscle fibers. Each of these muscle fiber has got myofibrils like this, arranged like this. I'm taking a cross section of this muscle fiber. All right. So they are composed something like this. And this forms a muscle spindle. This entire muscle fibers will form a muscle proper. This muscle proper, from outside it is covered by a T-tubule and terminal cisternae. The T-tubules are nothing but extensions of surface membrane extensions. Whereas the terminal cisternae, what happens is the sarcoplasmic reticulum that is part of internal membrane. This its important function is to store calcium. So in the skeletal muscle, the calcium is stored in sarcoplasmic reticulum. It's an important component. It's an important cell organelle. Sarcoplasmic reticulum. This sarcoplasmic reticulum, the calcium is stored in the terminal cisternae, which is close to the T-tubule system. Right? So let us see the ultrastructure of these each resting muscle fiber. See, we have got actin filaments like this. These actin filaments have got an important protein called as tropomyosin. So this is tropomyosin. The tropomyosin and the actin filament. In between these actin filaments, we have got an important protein embedded, and this is the troponin. Troponin proteins are of different types: troponin C, troponin T, troponin I. This troponin is very, very important in case of myocardial infarction. The serum levels of troponin are increased in case of myocardial infarction, and this is how you can diagnose heart attack. I'll discuss that when I'll be discussing general medicine. All right. Each of these troponin has got a calcium binding site. So whenever calcium comes and attaches to the troponin, it stimulates the it starts or begins the excitation contraction coupling. Excitation contraction coupling. So we have got actin filament, tropomyosin, troponin. All these are important filaments. What is actin filament? I already told you that actin is a thin filament or a thick filament. Actin is a thin filament. So actin is a thin filament. It has sites for the attachment to myosins, myosin binding sites. All right. Next comes the tropomyosin. Tropomyosin, it blocks the myosin binding sites on actin. What is the function of tropomyosin? It blocks the myosin binding site. It blocks the myosin binding site. Myosin binding site. Tropomyosin. Next comes the important protein called as troponin. What is the function of this troponin? Troponin, basically the function is it provides calcium binding site. So I told you there are three types of troponin: troponin T, troponin C, and troponin I. Troponin T, it binds to the troponin T binds to the tropomyosin. Tropomyosin. Next comes, under resting condition, there is no calcium attached to the troponin. But the troponin C provides calcium binding site, hence the name troponin C. In resting state, it is free. But in case of contraction, once the contraction begins, the calcium binding site opens. I mean, it provides a binding attachment to the calcium. Whereas we have got troponin I, which binds to the actin and inhibits the contraction. I for inhibition. So it binds to actin to inhibit contraction. To inhibit contraction. So important function of troponin I is to bind to actin to inhibit contraction, hence the name I. So when calcium binds to troponin C, the troponin tropomyosin complex moves, exposing the actin filament binding for the myosin. So what is the function of tropomyosin? It is blocking the myosin binding site, is it not? It is blocking the myosin binding site. So whenever calcium comes and attaches to troponin C, what does it do? When calcium binds to troponin C, the troponin tropomyosin complex moves, exposing the actin binding site, that is for myosin, that is the myosin binding site. All right. So I repeat again. So presence of calcium on troponin C, this causes the tropomyosin complex to move. So this complex moves and thus exposes the myosin binding site on actin. Thus the contraction begins. So this process will again try to analyze it in very much detail. What are the proteins present on the thick filaments? Please remember the myosin. The thick filament, that is the myosin, it has got a very important enzyme activity. Myosin has got ATPase enzyme activity. So myosin has got ATPase activity. So the splitting up of ATP puts myosin in a high energy state. Because it has got ATPase activity, so it can expenditure of this ATP, that is the energy resource. It makes the myosin an energy rich protein, and it increases the myosin's affinity for actin. Once the myosin binds to actin, the energy that is transferred to the chemical energy is converted into mechanical energy, causing the myosin to pull the actin filament and thus generate a power stroke. All right. And if there is any force generated in the power stroke, the muscle shortens. This results in isotonic contraction. So in this power stroke, if a force is generated, this is transmitted onto the muscle as a load, and then the muscle shortening occurs. If the force generated is not sufficient to move the muscle, if the force generated is not sufficient to generate the movement of the muscle or transformation of that into a load, then the isotonic contraction does not occur. Here there is no change in the muscle length, the muscle does not shorten, but still contraction is occurring. The muscle does not shorten, but still the contraction is occurring. Such type of contraction we call isometric contraction. I repeat. So first, what happens? Because of ATP activity, the energy is generated in the myosin. Because of this high energy state, the myosin pulls the actin towards itself to generate a power stroke. This power stroke, if it generates enough force to transform it into load onto the muscle fiber, that results in shortening of the muscle. Any contraction which causes shortening of the muscle, you are calling it as isotonic contraction. Sometimes the power stroke cannot generate enough force. If the enough force is not generated, it results in contraction, but there is no shortening of the muscle. Iso means normal or no change. All right, metric means muscle length. So if there is no change in the muscle length, it results in isometric contraction. Isometric contraction. All right, let us try to understand what are the chemical and mechanical changes that occur in excitation contraction coupling. Excitation contraction coupling. All right, let us try to understand. What I mean to say is there is a cross bridge. Let us try to understand what happens at this cross bridge. The cross bridge mechanics, let us try to understand. So it is very important to understand that it is the ATP and use of ATPase enzyme and hydrolysis of ATP generates energy. Because of this energy, the resting muscle suddenly becomes active and there is contraction. All right, but how is this all occurring? Let us try to understand. Please remember that ATP is not required to form the cross linking bridge to actin, but it is required to break the link with the actin. So please remember that ATP is required to break the link with actin filament. It's a very important statement you'll have to understand. The cross bridge formation, the cross bridge cycling, that results in contraction of the muscle. This continues in the following ways. Either there is withdrawal of calcium resulting in the contraction to stop at one position, that is the sudden withdrawal of the calcium results in muscle coming to the normal position, or this is one thing, that is withdrawal of calcium, or the second point is if the ATP is depleted, if there is no energy to form the contraction pathway, contraction linking, then also the contraction stops and the muscle comes back to normal position. Please remember that the contraction cycling stops in case of withdrawal of calcium, or in case of if the ATP is depleted. The second activity usually occurs when does the ATP deplete? At the time of death of the muscle fiber. There is no ATP, that results in sudden stoppage of that contraction coupling. All right, this is how these are the two the mechanisms of contraction coupling. So what is this coupling and what are the cross bridges? Let us try to understand with the help of a simple diagram. So please remember that, say suppose we have got the actin filament and the myosin filament, is it not? Actin filament and the myosin filament. So say suppose this is the myosin filament, and it has got a site for, and say suppose this is the thin filament, the actin filament. All right, so what happens is, because of ATPase activity, this is the first step, ATPase activity of the myosin. This puts myosin into high energy state. So high energy state of the myosin. This puts myosin into the high energy state, and this results in high affinity of myosin to the actin. In case of resting muscle, we all know that the tropomyosin covers the actins, the myosin binding site, I told you. So once the calcium enters, the calcium attaches to the troponin C. This exposes the myosin binding site of the actin. Thus, so say suppose this is the myosin filament, and this is the cross bridge. Thus what happens, now the actin comes in contact with myosin. And thanks to the calcium binding to the troponin C, which has increased the affinity of myosin to the actin. Once there is binding of myosin to the actin and myosin combine, and what will happen is the chemical energy here is converted into mechanical energy. So how is this happening? So this is the second step of myosin binding to actin. Now let us try to understand the third step. In the third step, what happens is there is energy transfer or energy change, wherein this chemical energy that is supplied by the ATP is suddenly pulls the actin. The myosin pulls actin. Myosin pulls actin to create what is known as power stroke. The actin filaments slide over the myosin filament. To produce active tension. I told you that if this power stroke is enough to create a load onto the muscle, the force is enough to create load onto the muscle, it causes contraction. Such contraction we are calling it as isotonic contraction. If the force is not enough to create a load onto the muscle, there is no shortening of length of the muscle fiber, but still contraction is occurring in between actin and myosin filament. You are calling it as isometric contraction. After that, the fourth step is the once the ATP is lost, the myosin becomes completely, once the ATP is lost, the myosin is not having any energy state. Now it has come down to low energy state. Hence, because of loss of ATP, that is used of ATP, not loss, but using up of ATP, there is dissociation or bringing back to the resting muscle state of the actin and myosin. Remember, I repeat, ATP is not required to form the cross-linking. Please remember this point. ATP is not, if you see this diagram here of myosin and actin, ATP is not required for cross-bridge linking. Please remember this point. But it is required to, what is ATP doing? ATP is required to break the link with the actin. Break the link and cross-bridging, that is contraction, continues until either of the two mechanisms, that is either withdrawal of the calcium or depletion of the ATP. How is this calcium? Withdrawal of calcium, I'm telling you activation by calcium. Calcium enters the cell. Calcium is withdrawn from the cell. How is this calcium entry, calcium withdrawal? Calcium entry, calcium withdrawal is occurring in the cell. To understand this, let us try to understand the regulation of calcium in the skeletal muscle. Let us try to understand this concept. Please remember that there is a cell organelle called as sarcoplasmic reticulum, which is a storehouse of calcium. Sarcoplasmic reticulum is a storehouse of calcium. It has got high concentration of calcium. There is a strong electrochemical gradient that is produced for calcium. So very strong electrochemical gradient is required for the calcium to come out of the sarcoplasmic reticulum and enter into the cytoplasm. There are two key receptors involved in the influx of calcium. There are two key receptors which are involved in the influx of the calcium from the cytoplasmic reticulum to the cytoplasm. The sarcoplasmic reticulum to the cytoplasm. And these two receptors are dihydropyridine receptor and ryanodine receptor. Dihydropyridine and ryanodine. These two receptors are very much important for entry of the calcium from the sarcoplasmic reticulum into the cytoplasm. The DHP receptor, please remember, it is a voltage gated calcium channel that is located in the sarcoplasmic membrane. So it is a calcium channel protein, which is located where? In the sarcolemmal membrane, that is membrane of the sarcoplasmic reticulum. Class gentlemen, John the John. Sarcoplasmic reticulum is surrounded by sarcolemmal membrane. So on this membrane we have got the dihydropyridine receptor. This dihydropyridine receptor is calcium voltage gated calcium channel, and the calcium does not flux through this receptor in the skeletal muscle. Rather, the dihydropyridine functions as a voltage sensor. Actually, it functions as a voltage sensor. Whenever the skeletal muscle is at rest, the dihydropyridine blocks the ryanodine receptor. So at rest, whenever skeletal muscle is at rest, it blocks the ryanodine receptor. All right. What is the function of ryanodine receptor? Let us try to understand. Ryanodine receptor is a calcium channel. Please remember that dihydropyridine is also calcium channel, voltage gated calcium channel, but it is not causing movement of calcium through it. In fact, it is not causing any movement of calcium through it, but it is functioning as a voltage sensitive receptor. Whereas ryanodine receptor, which is regulated by the dihydropyridine receptor, whenever muscle is at rest, but thus the calcium is prevented from diffusing out into the cytoplasm. So what will happen if the muscle is at at rest? The calcium is held back. We hold the calcium back into the sarcoplasmic reticulum thanks to the ryanodine receptor present on the sarcoplasmic reticulum. So what is the sequence? Let us try to understand. So skeletal muscle action potential is initiated first at the neuromuscular junction. From there, what happens? The action potential travels down to the T tubule. I told you there is one T tubule, that is the outermost membrane of the outer membrane called as T tubule. I told you here. So somewhere here, the outside covering of the muscle like this, we have got T tubule. So once the action potential comes to the T tubule, from here voltage changes occur. These voltage changes are sensed by which receptor? Voltage changes are sensed by dihydropyridine receptor, the voltage gated calcium channel. And once there is voltage change, the ryanodine receptor blockage by the dihydropyridine receptor, the ryanodine receptor blockage is lost. Once it is lost, removal of the DHP block allows calcium to diffuse into the cytoplasm. Once the calcium diffuses into the cytoplasm, it binds to the troponin C. Once it binds to the troponin C, the troponin tropomyosin complex is moves and it exposes the myosin binding site. There is rise of cytosolic calcium. This opens up the ryanodine receptor channels and thus calcium is released. All right, thus calcium is released. So calcium releases into the cytoplasm. Now what's happening is this calcium binds to the troponin C. Because of this, I told you already that the cross bridge cycle gets initiated. The cross bridge contraction coupling cycle gets initiated. The cross bridge gets initiated. Now, once the calcium binds to troponin C, it initiating calcium, the cross bridge, and once the active tension is created, now calcium has to go back to the place from where it has come. From where is it, calcium has come? The calcium should go back to the sarcoplasmic reticulum. How does it go back to the sarcoplasmic reticulum? By a calcium ATPase protein called as sarcoplasmic endoplasmic reticulum calcium ATPase channel. It's a very, very, very important point. All right. Let me show you this. Let me show you this with the help of a simple diagram here. So are you able to follow this diagram? Hope you are able to follow this diagram. It's a sarcolemma. I repeat again all the important steps which I am showing you. These steps are one is action potential reaching the sarcolemmal protein. This is the first step. Point number 1 here. Point number 1 here. If you are able to follow, that is onto the T tubule. From the T tubule, this action potential change is causing release of calcium from the sarcoplasmic reticulum. So this is the sarcoplasmic reticulum. We have got ryanodine receptor. This is the ryanodine receptor and this is the DHP, dihydropyridine receptor, which is a sensor of the voltage change. Who senses the voltage change? It is the dihydropyridine receptor. Dihydropyridine receptor, it senses the voltage change. Dihydropyridine receptor senses the voltage change. This voltage change is results in removal of the blockage on the ryanodine receptor. Ryanodine receptor releases calcium from the sarcoplasmic reticulum. So this is the calcium release pathway. So this is the calcium release pathway into the cytoplasm. This calcium goes and binds to the troponin C. From troponin, it causes movement of troponin tropomyosin complex, exposure of the myosin binding site, contraction coupling actin and myosin combine to each other, contraction excitation complex is started, and muscle contraction occurs. Once the work is done, calcium should be pumped back to the sarcoplasmic reticulum, and it is done by the sarcoplasm endoplasmic reticulum calcium ATPase protein, SERCA protein, SERCA, SERCA protein. Hope you are able to. So whenever there is fall in the levels of cytoplasm, calcium in the cytoplasm, this causes the tropomyosin to again go back and bind to the troponin. This causes tropomyosin to again go back and bind to the troponin. Again, the actin binding site is covered, the myosin binding site is covered on the actin, and the muscle relaxes. Right? So what are the important points here? There are two ATPases. There are two types of ATPase mechanisms involved here. The first ATPase, I told you, it is a myosin ATPase, is it not? I told you that there is activity of myosin ATPase. Just now I told you. So here I told you that there is activity ATPase. This is the first ATPase activity that we have learned, the myosin ATPase activity. What is the function of this myosin? It supplies ATP rich energy, chemical energy converting into mechanical energy in myosin. So this protein, this ATPase activity is a specific to protein myosin, the thick protein called as myosin. Thus myosin becomes high energy state thanks to the ATPase activity of the myosin. The second important ATPase activity is SERCA, SERCA, which pumps back calcium into the sarcoplasmic reticulum, and this is occurring at the level of the cell organelle. That is the this diagram I have already explained you. So these are the two important points you will have today's take home message to understand the excitation contraction coupling. All right. Let us try to understand what are the mechanical response to a single action potential. Whenever a single action potential occurs, there is release of calcium. Release of calcium causes muscle twitch. Release of calcium causes muscle contraction or muscle twitch. The muscle membrane, which is completely repolarized, so once the twitch is over, the muscle membrane gets repolarized. So initial depolarization followed by repolarization. Whenever there is gain of charges by the muscle, you call it depolarization. All right. And whenever there is loss of charges, bring back to normal polarized state, it is called as repolarization. All right. These are the basics of physiology. I'll explain you. Say suppose this is one cell, some cell or muscle cell or any cell of our body, cell with a cell membrane. Any cell has got inside negative charges and outside positive charges. So this is called as polarized state. Polarized state. What happens is whenever an action potential comes and strikes the muscle membrane at the level of neuromuscular junction or somehow, what happens is this polarized state is lost. We call it depolarization. Depolarization. That is, the positive charges will enter into the cell. This is called depolarization. Once the depolarization wave is gone, it occurs for a short period of time. It has got a purpose. The purpose of depolarization in muscle is to cause muscle contraction. So once this depolarization is over, then again the muscle comes back to the normal polarized state. We call it repolarization. So gain of charges, depolarization, loss of charges, bringing back to normal polarization. You are calling it as repolarization. This is the basic of physiology you will have to understand. This concept helps us to understand the ECG also in case of cardiology. Again, if I teach you ECG, I'll explain you this concept again in detail. All right. So that helps us in understanding of the muscle fibers. We have understood various actin myosin interactions. We have understood how calcium is released. All right. Please remember that for the regulation of the regulation of skeletal muscle that we have talked. Regulation of smooth muscle is slightly different. What happens in regulation of smooth muscle is that if you talk about the smooth muscle, what happens is in the smooth muscle we have got voltage gated calcium channels, and these calcium channels are L type of calcium channels. The smooth muscle calcium channels, L type of calcium channels that reside in the sarcolemma of the smooth muscle. All right. There is depolarization. Whenever depolarization occurs, it opens up these channels. Depolarization opens calcium channels. It undergoes calcium release into the cytoplasm. From where? From same sarcoplasmic, from the sarcoplasmic reticulum. From the sarcoplasmic reticulum. So here calcium induced release of calcium. So small amount of calcium is coming, it is stimulating the release of more calcium. So this is an important property of smooth muscle, different from skeletal muscle. In the skeletal muscle, it was a depolarized state, the action potential of the T tubules which was causing the release of calcium. But here small amount of calcium comes and it opens up the channel. Because of this small amount of calcium enters in the sarcoplasmic reticulum to release more calcium. All right. There is one more pathway by increasing the inositol triphosphate, IP3 increase. Whenever there is increase in the IP3, this also causes calcium efflux from the sarcoplasmic reticulum. IP3 is increased whenever there is G protein coupled reaction. G protein coupled reaction. G protein is a second messenger system which we already discussed in endocrinology, and that was responsible for release of calcium from the sarcoplasmic reticulum and to cause the muscle contraction. So the cytoplasmic calcium, it binds to an important protein. So the cytoplasmic or cytosolic calcium, it binds to an important protein called as CAM protein or calmodulin. Its name is calmodulin. We are talking about smooth muscle, not skeletal muscle now. Calmodulin. This calmodulin, that is a calcium calmodulin complex, it activates the myosin light chain kinase. This myosin light chain kinase is an enzyme which phosphorylates the myosin light chain. So it has got a phosphorylation activity. Phosphorylation activity. Because of this phosphorylation activity, the myosin light chain is stimulated. Hence the myosin binds to actin. Once there is myosin binding to actin, results in contraction of the smooth muscle. Contraction of the smooth muscle. So whenever there is increase in cytosolic calcium, the myosin light chain kinase is stimulated. Myosin light chain kinase causes muscle contraction. The smooth muscle, where whenever there is exactly opposite, whenever there is loss of calcium, what will happen is the this process undergoes in a reversal fashion. Please remember that smooth muscle reduces cytosolic calcium by the same mechanisms as the cardiac muscle. Same mechanism as the cardiac muscle. Cardiac muscle mechanics. We'll quickly discuss cardiac muscle mechanics. We'll try to understand cardiac muscle mechanics here. So skeletal muscle and cardiac muscle, if you compare, cardiac muscle and skeletal muscle, both are striated muscles. But what are the differences? What are the important differences? So cardiac muscle versus the skeletal muscle. Let us point out the differences first. Please remember that cardiac muscle has got gap junctions. These gap junctions are important for, say, suppose this is one cardiac muscle cell, this is another cardiac muscle cell, this is another cardiac muscle cell, this is another cardiac muscle cell. It has got gap junctions. So these gap junctions are responsible for transfer of charge from one cell to another cell. Hence, one cell to another cell is stimulated in a linear fashion. So these cells are stimulated in a linear fashion. So say suppose you stimulate the first cell, the charges transfer occurs in the next cell, then the charge transfer easily occurs in the second cell, third cell, then the fourth cell. So this is an important difference between cardiac muscle and skeletal muscle, where skeletal muscle do not have any gap junction. Second is the sarcoplasmic reticulum calcium. The sarcoplasmic reticulum related calcium, the sarcoplasmic reticulum calcium can be altered in case of cardiac muscle. It can be easily altered in case of cardiac muscle because of the ATPase activity, because of the sodium potassium, that is the ATPase activity. There is one channel. I'll discuss this when I'll be discussing heart failure chapter in medicine. Again, I'll remind you about the differences and similarities between the cardiac muscle and the skeletal muscle. It's an important discussion now. Please remember that extracellular calcium is very important to cause cardiac muscle contraction. So this is the first point, this is the second point. Third point is extracellular calcium. Extracellular calcium means calcium from outside. So if this is the cardiac muscle cell, calcium from outside can enter into the cardiac muscle and can stimulate the release of calcium from the sarcoplasmic reticulum, resulting in contraction of the cardiac muscle. So extracellular calcium can induce calcium release from the sarcoplasmic reticulum in case of cardiac muscle, but this is not a property that is present in the skeletal muscle. The cardiac muscle, how does it remove cytosolic calcium? Removal of cytosolic calcium by cardiac muscle is by two mechanisms. So fourth difference is, so the removal of calcium from the cytoplasm in case of cardiac muscle, it is brought about by two mechanisms. One is the sarcoplasmic endoplasmic calcium ATPase protein, the SARCA protein. One is the SARCA protein. Another is the sodium calcium channel. The sodium calcium channel exchanger, we call it. Sodium calcium exchanger channel. Another is the sodium calcium exchanger channel, which is present in the sarcolemmal membrane. The skeletal muscle has got only SARCA, but it has not sodium calcium exchanger channel. Is it not? The sodium calcium exchanger channel is not there, but in myocardial cells it is there. The cardiac cells have got a prolonged action potential. Cardiac cells will have prolonged action potential because of the movement of potassium into the cell, all right, which maintains the positive charge. So action potential, in our next class we will be discussing what are the differences between action potential, but for now please remember the myocardial cell has got a prolonged action potential, whereas skeletal muscle has got a quick action potential. So skeletal muscle has got action potential something like this, whereas the cardiac action potential is something like this. It has got a prolonged action potential, thanks to the gap junctions, thanks to the potassium channel which open up even during the depolarization state. Thus the positive charge is maintained. I'll explain you each and every point in my next class. So here, is the cardiac muscle different from skeletal muscle, or is there any similarities? Yes, they have few similarities. For example, the cardiac muscle has got the same actin, the same troponin, the same myosin filaments, the tropomyosin, the myosin, the troponin, and the titin. I remember titin just now in beginning of my class I told you the protein called as titin, T-I-T-I-N. So all these are same in both cardiac muscle and the skeletal muscle. So this is the point number one of similarity. Second point similarity is both have got SARCA protein for removal of calcium from the cytoplasm. Both have got ryanodine receptors. Ryanodine receptors are also common in sarcoplasmic reticulum, and they show calcium induced. Ryanodine receptor is for calcium induced release. It is a receptor present on the sarcoplasmic reticulum. Any rise in the cytosolic calcium begins the excitation contraction coupling, both in the cardiac muscle and also in the skeletal muscle. So rise in the cytoplasmic calcium, is responsible for excitation contraction coupling. These are all the important similarities. The last similarity in the cardiac muscle and skeletal muscle is the activity of ATP, which plays an important role in providing energy. So these are all the important differences and dissimilarities and similarities. These differences and similarities of cardiac muscle and skeletal muscle. You will have to remember all these. Right? I also told you about smooth muscle regulation. So this smooth muscle regulation completes the topic of the first topic of muscle physiology. So first topic of muscle physiology is completed. Let us move on to the next chapter. The next chapter, chapter 2 in this video, will be talking about skeletal muscle mechanics. Skeletal muscle mechanics. The skeletal muscle mechanics, you will have to understand what is preload, what is afterload, what are the white muscle fibers, what are the properties of red muscle fibers. All right. So let us try to understand. I already told you this when I was discussing cardiology, but here we'll try to understand with the help of certain examples. So skeletal muscle mechanics, we start with understanding of preload. The first topic of discussion will be preload. So this is chapter 2 of muscle mechanics. We'll try to understand what is preload. Please remember, one important simple fact is that any load on the muscle which is in relaxed state, you are calling it as preload. If muscle is relaxed, say suppose muscle is relaxed, just before contraction, the amount of load that is present on the muscle, you are calling it as preload. Why are you calling it as pre? Because it is just before contraction. And why are you calling it as load? Because it is the amount of load that is present on the muscle. So amount of load on the muscle before contraction or in a relaxed state. Can I call it before contraction? Obviously relaxed state. Please remember, whenever you apply preload to the muscle does two things. You apply the preload stretches the muscle, and preload generates a passive tension in the muscle. Please remember, this concept is important to understand. One, the cardiology. Second, first is cardiology, to understand cardiac mechanics. Second is to understand the tone of the muscle in neurology. In neuroanatomy or neurology, I shall be discussing again this point. But here I am giving you all the basics. So what are the two factors that, what are the two things that happen to the muscle whenever you apply preload? Whenever you apply preload, the muscle stretches. There is a stretch, muscle stretch. Muscle stretch is occurring. Second thing is a passive tension is generated. Passive, not active. Passive tension is generated. Passive tension is generated in turn. And we all know that muscle is elastic. Muscle is a elastic structure. Please remember. So what protein is responsible for elasticity of muscle? Just now I told you, titin. Titin is a protein which is responsible for elasticity of muscle. Just now I told you. Titin is responsible for elasticity of protein. Titin is responsible for elasticity of protein, is it not? So we all know that muscle is an elastic structure. So any elastic structure, it resists the stretch. Whenever you try to pull it, it will resist and it will come back to its normal state. This is what is elasticity. All right. When you try to pull a rubber band, what will happen? It will come back and it will hit you. So that is nothing but the elasticity. Who is giving this elastic nature to the protein? Which protein is giving elastic nature to the muscle? It is a titin. So please remember, the greater the pull, the greater will be the passive tension. So the passive tension is nothing but the elasticity. So when greater the pull, The greater the pull, the greater will the passive tension. Please remember this part. All right. Whereas any stretch on the muscle, the greater the preload, the greater will be the stretch. So it is directly proportional to the preload, stretch and passive tension. Now let us try to understand the afterload. Let us try to understand the afterload. Afterload is the load of the muscle that is applied onto the muscle and on which the muscle is working against it. For example, if you want to lift something, for example, if you want to lift a bottle, water bottle, all right. So whenever you're lifting the water bottle, the weight of this water bottle is acting as an afterload. The weight of this water bottle is acting as an afterload to lift it. All right. All right. So whenever afterload is applied, I explain you again this afterload. Afterload is nothing but it is the weight the muscle works against with. It is afterload is a load the muscle works against. All right. Against which the muscle is working. So whenever you apply afterload, what will happen is if the muscle generates a force equal to the weight that is applied, the muscle shortens. If the muscle is unable to generate the force, contraction occurs, but the muscle does not start shorten. I repeat. So the force that is generated by the afterload, if the force that is generated by the afterload moves the muscle, if it is enough to move the muscle, then the muscle shortens. This shortening of the muscle, contraction with shortening, you are calling it as isotonic contraction. I told you, any afterload which is equal to the force required to move the muscle, you are calling it as isotonic contraction. Say suppose this force that is generated, it is less, the afterload is generating less force to move the muscle. The muscle is not shortening, muscle is not shortens. The muscle does not shorten. This is termed as isometric contraction. Isometric contraction. What are the types of tension that are applied? We apply a passive tension onto the muscle, active tension. All right. Passive tension is nothing but the preload. Active tension is nothing but the afterload. Simple. So the tension or force that is applied, passive and active. Passive tension is nothing but the preload. Active tension is nothing but the afterload. So afterload can result in isotonic contraction or isometric contraction. The preload can result in a stretch. Preload can result in a passive tension, that is the elasticity that determined by the elasticity of the muscle fiber. So these are all the important muscle mechanics you need to understand before you understand the passive tension curve and the active tension curve, all these points. All right. Now we'll have to understand an important graph which comes in USMLE based exams. This is the length tension curve, we call it. Length tension curve. This you can apply it in any muscle, skeletal muscle in our body, and thus we may come to a conclusion whether it's a passive tension curve that is occurring or a active tension curve that is occurring. Please remember, passive tension means preload, active tension means afterload. I already told you this part, right? So the length tension curve is an important curve for understanding what both the skeletal muscle and the cardiac muscle function. Both the skeletal muscle function and the cardiac muscle function. So you try to draw a graph. You try to draw a graph, and this graph is measuring the tension that is applied onto the skeletal muscle or cardiac muscle, and you try to understand what are the changes that are occurring. What are the changes that are occurring? What are the changes that are occurring across this? What are the pattern of changes that are occurring in this graph? So please remember that, say suppose on the y-axis we have got tension, that is in grams, tension or weight that is applied in grams, and on the x-axis we check the muscle length in some units. All right, say suppose 1 2 3 4 5 6 7 8, something like that. 1 2 3 4 5 6 7 8 9 10. All right, here we have got, say suppose in grams, you are keep on applying, so increasing the weight 1 2 3 4 5. At one point of time, what happens is the muscle stretching starts, is it not? So let us try to understand when there is no preload. When there is no preload at this point, say suppose this is a point A, and from here the muscle stretch is starting slowly. All right, A is a point where there is no preload, thus the muscle does not stretch. There is no passive tension, the muscle does not stretch. With each one, say suppose 1 gram of increase in the weight, the muscle has come stretch here. Say suppose it has from 4, it has come to 6.5 or 6.1, something like that. So with each increase, the muscle is increasing its, with increase in the weight, say suppose you have come up to 5, and maximum length that is achieved is 9. So this maximum length that is achieved, you are calling it as point C. Maximum length that is achieved, you are calling it as point C. And say suppose somewhere point B, somewhere is point B. All right, what is this point B? Point B is nothing but 1 gram of force that is applied onto the stretch that is applied onto the muscle, resulting in some change in the muscle length. It has come to point 6. And what is point C? Maximum load that is applied. 5 grams of load is applied, producing the maximum change in the length and resulting in a great tension onto the muscle. All right. So this is about understanding of the passive tension. Passive tension. If you apply active tension onto this graph by stimulating the muscle, and result in muscle contraction at different preloads. So in this example, what we are doing is the contraction we are calling it as a maximal isometric contraction. I told you what is isometric contraction. Contraction is occurring, but it is not creating any change in the muscle length. The contraction produces tension, but there is no change in the muscle length. Thus the muscle does not shorten. So please remember that at a point of preload of A, actually what happens is, if you see the active tension here at point A, so somewhere from point 3, so the graph is coming like this, something like this, and at one point of time you see no further contraction. So how to explain this? Let us try to explain this. So whenever there is no preload, the muscle, say suppose, has got a contraction of at around 3. All right, this is active tension. that is, you have applied 3 grams of what do you say, weight onto the muscle. So this is the beginning point 1 of the afterload. So here muscle is already at one single length of 0.4 units somewhere here. All right, so there is no change. But as you increase the tension, say suppose at 4 grams of, if you are applying 4 grams of tension, the muscle has increased in its length. Now at some point of time you increase the weight, but the muscle is not increasing in the length. Instead, beyond a stretch point, what's happening is the muscle tension begins to fall. The muscle tension begins to fall, and this is because there is a relationship between the maximal length and maximum tension that is creating a contraction. So this is what is called as isotonic contraction. Isotonic contraction. This is what I want to explain. So from here to here, there was change in the length. But from this point to this point, there is no change in the length. So this is the isometric contraction. No change in the length. Isometric contraction. So this is how you will have to understand. It's an important concept, theoretical concept, but very few applications are there. So we have got actin filament overlap. If you combine this concept with the actin and myosin, if you combine this concept, we come to a conclusion that there are important points to be learned here. So same figure if you apply onto the sarcomere, you may come to a conclusion that, give me a second, just changing the settings here. So initially what happens is there is actin filament overlap which does not cause any change in the muscle length. But once the myosin binding site is exposed, the actin and myosin combine and it changes the muscle length. Then after that what happens is suddenly the calcium goes back into the sarcoplasmic reticulum, and the whatever contraction has occurred, it gets converted to relaxation, and again there is only isometric contraction phase where there is no change in the muscle length. So this is what is application into actin and myosin cross bridges. The number of actin and myosin cross bridges directly proportional to the tension that is created. Please remember, the number of cross bridges is directly proportional to the tension that is created. The active tension generated is greatest at one particular weight. The action potential, that the active tension that is created is greatest because the maximum overlap of actin and myosin has occurred. So any change, maximum overlap, so I'm saying that it is directly proportional. Directly proportional means what? Maximum overlap results in maximum change in the length. Maximum overlap results in maximum change in the length. But at one point of time there is a maximum stretch, that is whatever possible stretch is achieved. After that there is no active tension. Once there is no active tension, there is no change in the muscle fiber length. In fact, there is isometric contraction, that is contraction occurs but without change in the muscle, without causing any further muscle shortening or lengthening, whatever it is, without change in the muscle length. So this is what is, you will have to understand application of this graph, right? There are different types of fibers, the white fibers and red fibers. To conclude the topic today's topic, we have covered two chapters of muscle physiology, skeletal muscle mechanics, and then we also understood the excitation contraction coupling. So to conclude the topic, please remember that there are properties we'll have to understand. What are the white muscle fibers and red muscle fibers? So white fibers, why do you call it white fibers? So generally what happens is large powerful muscle is called as white muscle, and it is performing short-term activity. For example, eye, muscles of the eye, ocular muscles. So short term activity but powerful activity, very powerful muscle. Eye muscles are very powerful but short term activity. For example, in an athlete, the leg muscles, he'll, for example, a person called as Usain Bolt, his leg muscles, if you see, leg muscles of Usain Bolt, Mr. Usain Bolt, if you see, the famous sprint runner, the sprinter, they perform activity very fast. They're powerful muscles and performing activity very fast. What does that mean? I am talking about a large muscle per motor unit. Per motor unit. This large muscle has got high ATPase activity. Hence, rapid contraction, relaxation, contraction, relaxation, contraction, relaxation. It has got very high aerobic glycolysis pathway, so that ATP is generated and giving back to the muscle for contraction. And most importantly, it has got low myoglobin. So these are all the important properties of white fiber, the large muscle fiber which has got rapid activity, large muscle fiber which has got rapid activity. Exactly differences you can see in the red fiber. So what are red fibers? What are the red fibers? These are the small muscles, less powerful muscles, usually for endurance, long term. For example, you are standing for a very long time. From morning till evening, a policeman has to stand on the road, or policeman has to stand guard, security guard has to stand outside one apartment. So it is to maintain the body posture for a very long time. We require certain endurance, long-term muscle activity. So small muscles which perform long-term muscle activity. Long-term muscle activity you are calling it as red fiber. What are the important characters of red fiber? It has got a small muscle per unit. It has got low ATPase activity, exactly opposite. It has got high capacity of aerobic metabolism because of changes in the mitochondria, and it has got very high rich in myoglobin protein. This myoglobin protein is giving the red color. Hence we are calling it as red fibers. So these are the differences between the large fiber, that is a white fiber, and a small fibers, that are the red fibers. Small fibers are red fibers, large fibers are white fibers. Low myoglobin and high myoglobin make it white and red. So thank you very much for patient listening. Hope this class was quite long. The graph aspect was slightly difficult to understand. Application part, you can easily understand it once you apply it in the actin myosin excitation contraction pathway. Thank you for patient listening. If you have any doubt, please get back to me and I'll be happy to help you out. Thank you very much. Good night.